Hello and welcome to the Nobel Spirit session. My name is Ashley Colburn and I'm honored to be joined with 3 Nobel Prize laureates at the 10th Annual International Society for Applied Biological Sciences Conference in the beautiful Rector's Palace in Dubrovnik, Croatia. We'd like to send our regards to all of those tuned in on Croatian National Television and our European Broadcasting Union members. Never before have intellectuals from 42 countries gathered together for a public discussion on the role of science in solving global health issues. We have also invited the brightest students from around the world, and later in today's panel, they'll be addressing our laureates with questions and comments. So, among us today is the science elite, and we're going to explore how their discoveries have changed our world we live in. First, I'd like to introduce our Nobel Prize laureates. Professor Ada Yonat from Israel, who was awarded the Nobel Prize for studying the structure and function of the ribosome. Professor Robert Huber from Germany, who determined the three-dimensional structure of a photosynthesis reaction center. We have Professor Harold Zurhausen, who joins us from Germany, who discovered that human papillomavirus can cause cervical cancer. And finally, Professor Dragan Primorac, the president of ISOBS. Thank you all for being here today. Also, we'd like to get to know these laureates and a little bit more about their discoveries. So, Professor Yonat, we're going to start with you. After studying the ribosome for so many years, can you explain to us the importance that ribosomes have on the body?
They are most important. Ribosomes exist in every living cell, from bacteria, cockroaches, flowers, trees, human, elephants. And they do a very important, basic, fundamental task. They are translating the genetic code, the instructions in the genetic code, into the workers of the cells, which are called proteins. They are doing it all the time. Every time that a protein is needed, which means continuously, and they exist everywhere. Without them, there is no life. Is this enough?
Yeah, but can you tell us that when will we be able to fully understand ribosome structure and stop bacteria from growing antibiotic resistance?
Well, now you ask 2 questions. I think that we understand the ribosome structure at least in terms of their function, because that's the most important thing. If there are other regions in the ribosome that we understand, but we don't know why they're there, we are still working on it. Ribosomes are large. It means by eye we don't see them. They are so small. They are in the cells, but within the cell they are the largest particles. So they are made of many, many components, 4,500 nucleotides. They are made of 2 types, nucleic acids like RNA and proteins. And in human, 80 different proteins. So there is something to discover and understand. But what we understand now, which in my opinion is the goal of our studies, is how they function, how they decode the genetic code, and how they produce the proteins accordingly, how they don't make mistakes or hardly make mistakes. Mistake 1 to a million, which is hardly anything. Maybe they make more mistakes, but we cannot even catch them. When they have a mistake, they get rid of it before we human beings can catch it. Now, because they are so important in every living cell, also in bacteria that cause diseases, pathogenic bacteria, many antibiotics are made to hamper them, to paralyze them. About half of the useful antibiotics are paralyzing ribosomes, or at least paralyzing the process. So understanding the structure and where the antibiotics are working, where in which part of their structure, in which function, in which task within the function, we can understand how antibiotics work and maybe make them better or fight against resistance. This is what we are doing now.
That's a fantastic discovery. Now, Dr. Huber, you've been researching proteins of the immune system during your lifetime. Now, do you think that the human body and its DNA code has the potential to develop resistance to different diseases the way that simpler organisms can?
That's a long question, but I thought that I'm asked, what did I do for my Nobel Prize?
Yes, I mean, go ahead and expand a bit on that too, because it's very complicated.
This I like to answer saying that it was on photosynthesis, to understand photosynthesis. Now, photosynthesis is the basis of all higher life. Plants, we eat plants, we eat the animals that eat plants, and plants and also photosynthetic bacteria make material, make the stuff that we are able to eat or to use by using the sunlight. How is biology dealing with this problem? We do know that we have the technical photocells made from silicon, for instance, or using electrical metal wires. All of these materials is not available in nature. Nature has proteins and some cofactors, for instance, the chlorophyll cofactors, the green color of the plants. So how does that work? And that is what we wanted to find out by isolating the biological photocell and crystallizing it. This is a long story, how you get from crystals to molecular structure, and then determining the molecular structure in atomic resolution. So that was the basis of the Nobel Prize, which I shared with two colleagues. But it was, of course, based on the development of the technique, the crystallographic technique, and that is a long story that goes 100 years back, to develop a technique of having a super microscope with which you can resolve in atomic detail the structure of large molecules, which is a technique that now can be used for medicine, and Ada spoke about the ribosome that also has been analyzed using X-ray crystallographic techniques, but many other medically relevant biological molecules are analyzed by this technique. So it helps medicine, it helps pharmacology, it helps mankind. Not only that we know the physical and chemical principles of a biological process, but that we also can make use of it for medicine.
Now, you've been studying this for almost a lifetime, but when you decided to go into research, you grew up during World War II in Germany. I mean, is this when you started to really read the books and know that you were going to have a career in science?
Well, I knew also as a high school student that I would study chemistry. That's what I knew. And then I specialized on crystallography because I was fascinated by minerals. I lived and went to school in Munich, close to the mountains. So when you walk in the mountains and have some open eyes, you see these wonderful crystalline minerals which I collected. And then when I studied chemistry, I wanted to work in that field, to find out what makes crystals so as we see them.
And it all worked out. here we are
well,
today.
it all worked out, yeah. For me, it was quite straightforward. Yeah.
Now, Professor Surhausen, you studied HPV for 10 years. Now, when did you first realize that this could perhaps save millions of lives?
Well, I studied it even for 30 years, so for quite a long period. I'm afraid to say that I'm working since 55 years in this field. But indeed, we worked initially on a different type of virus and its role in tumors, Epstein-Barr virus in specific childhood tumors and also in some tumors which occur in Southeastern Asia. And subsequently, during this period, I found reports that genital warts, which are clearly linked to papillomavirus infections, very rarely convert into malignant tumors. And that triggered the idea, well, if that is the case, then at another site, namely at the cervix, they might produce something else as well. So for quite a number of period of time, we started to isolate and purify the viruses from genital warts, quite unsuccessfully in the beginning, but what we saw was that there is not the human wart virus, which everyone knew at that time was one virus, but there are at present more than 400 different types infecting humans. And after a couple of years, we finally were successful in isolating, purifying the virus from genital warts. Didn't find it in cervical cancer, which was a disappointment, but with another isolate obtained from the same site, we were more successful and could demonstrate those agents which today represent about 70% of those agents which are found in genital cancer. This induced a very rapid development subsequently, and also the development of vaccines, which are now widely applied to people prior to the onset of sexual activity, in the initial phase mainly to girls. Right now we advocate very strongly the vaccination of boys, claiming that if we would only vaccinate boys, we probably would prevent more cervical cancer than only by vaccinating girls. The reason is an obvious one, because in the age group, let's say between 15 and 40 years, almost everywhere in the world, the males have more sexual partners than the females of the same age group. And so for those reasons, they are the transmitters really of the virus quite effectively. And I even particularly in view of the fact that there are a number of young people here, I would kindly ask you consider this seriously, to vaccinate boys at the age between 9 and 14, certainly as well as girls. It's an important issue. Now, we continue to work on this for quite a while, studied the mechanism by which these viruses cause human cancer or are linked to human cancer. So we could explain the long latency period between infection and the occurrence of cancer, which is 15 to 30 years in cervical cancer, and also studied a number of other systems. So after finishing this whole time period, it was about at the turn of the century, we shifted to other systems and now working in different types of cancers and infections, possible role of infections.
Now, on a more personal level, in some of your free time, I know I've read that you enjoy photographing animals and traveling. Now, would you say that studying the behavior of animals or anything has impacted your discoveries in the medical world?
Well, even as a very young boy, I was fond of the fauna and also of the flora. And now, since my wife is South African, we are frequently traveling to South Africa, having safaris there. And I'm enjoying it tremendously. I'm really an ardent animal photographer. I'm not so sure whether it very much influenced my work, but let's say, for instance, quite in contrast to many of my medical colleagues, I almost never worked with experimental animals. So I worked always with humans and human materials.
Yeah, because I feel like a lot of times when people are doing research, they are working with the animals.
That's true. And it's, of course, for some it's essential. I felt for us it wasn't essential. It was important to get access to clinical materials and to analyze them directly for agents which are potentially involved in human cancer or other diseases.
All right, well, thank you. Now, Professor Primorac, first of all, thank you for bringing such outstanding individuals to today's Nobel Spirit session. Now, ISEPS has been around for 20 years. This is the 10th annual conference. Can you just tell us the importance of this conference, and especially specifically this year, what are some topics of discussion that you feel really stand out?
Thank you. The success of the conference is basically linked with the people who are coming. So Professor Ada Yonat, Professor Surhausen, and Professor Robert Huber, and many other people. including Mayo Clinic as a leading institution in healthcare business in the United States, St. Catherine Hospital, our strategical partner, Philips. So when you see how strong we are going in order to apply the new advantages in the field of the medicine, it's very clear why our focus is on the personalized medicine on one hand, on forensic genetics on the other hand, and on the third part is anthropological genetics. So since Professor Vuk Pavlović and Moses Schenfield, myself, started this series of the conference, we are basically handling those topics, I would say, from the best possible perspective with the clinical application. For example, we would like to see how we can apply all this knowledge, what we discussed today, in the clinical practice. What to do with stem cell therapy. And I witnessed today, and I told you that we are having first clinical trial at St. Catherine Hospital in order to treat the cartilage. We would like also to see what is the effect of the microbiome. And it was outstanding lecture today Professor Ada Yonat gave. You know, human papillomaviruses. One discovered change entire field. I mean, if you see what is happening and what is the percentage of the people that are going to die by 2050, according to statistics, it will be more than 10 million. So actually, more people will die from the bacteria than from the cancer. So what we are trying to do, we are trying to find some answers. We are trying to bring best people from the field. And once again, I would like to thank all friends coming to Croatia. Croatia is today the place where I would say the most famous people with great knowledge are trying to witness the future of the medicine. And that's what is ISEPS about.
And also we have a lot of young people that joined the conference as well. I mean, what is your goal behind that? Yeah. Do you feel like there are enough resources out there for these future scientists that maybe perhaps they could be sitting up here one day also as Nobel Prize winners? Yeah, What's out there?
It was very clear from all our professors sitting here that they would like to spend most of the time with the students. I'm very happy to tell you that traditionally we are awarding by Young Investigator Award top students, actually PhDs, under 40 from all around the world. So that's very, very prestigious award. And this year, for the first time, we are awarding actually 6 high school students who decided to apply to write essay in the field of molecular biology and genetics. And I'm so happy and proud to see all of them here and their mentors as well, because they're responsible for this work. And actually, that is what is life about. There's a new generation. And what I said to our students, those colleagues, physicians, students of the medicine today, they'll be the group that are going to apply the field of the personalized medicine as we are dreaming today. We would like to tailor right therapy for the right patient at the right time. Every year, at least 150,000 people in the United States dying because we don't understand pharmacogenomics. About 2.5 million people ending in the hospital because of adverse drug reaction. And those people that are having huge responsibility to protect us from this kind of the things in the future.
Now, one thing I want to have a discussion about with all of us, and especially because we have so many aspiring scientists here today. I mean, Professor Serhausen, when you started your scientific career, you didn't really have maybe so much support. How are you giving back to young people today?
Well, I had a lot of support, I must say, because even when I started to work in microbiology, I came to an institute where I had a lot of financial support, but I got no advice. For 3 years, I was totally on my own, had a number of crazy ideas. Some of them were really crazy, some of them worked out to some degree, and I wish today that some of the young people would have this spirit too, because for me it was the most helpful one. But subsequently, of course, where I went for a couple of years to the States and subsequently returned to Germany, I was relatively well funded throughout my life. I must say, in Germany we had the German Science Foundation, the Deutsche Forschungsgemeinschaft, which was quite helpful. And apparently my peers believed somehow that the studies would be fruitful in the end. I don't know why, but it is obviously true, so I have never had to complain about a lack of funds.
Mm-hmm. Now, Professor Yonat, would you say that obviously there's challenges along the way too? What were some of your challenges when you were doing your research that, you know, I mean, obviously you sit here with one of the most prestigious awards in the world, but I know that it wasn't easy probably to get here.
So first of all, I want to answer or to relate to a question that you asked, Robert, but I didn't want to disturb. You said almost your life or lifetime project. He's still alive.
But
So
you had, yes, exactly. But you are the most experienced here, as you've had your award the longest.
Yes, that's true. I'm in that sense.
And still researching, I'm sure.
The eldest, almost 20 years earlier, I got my prize. So I had to live with that prize for quite a long time. I had problems first. It was with the prize. It was stressful, I think. But that started to become less and less important, and now I enjoy it. Also because my contacts with young people becomes much easier, and I still do have a very small research group many years after my retirement, because of my connections I had with pharma industry. They support me and my small group. This I enjoy, and I would not like to miss contact with experimental work, because it keeps you on the ground. You see, otherwise I could spend all my time in traveling and lecturing and losing then contact with this research reality. The research reality is a failure rate of, I would say, 90% or 95%. So we live on this small margin of 5%. So experimental work keeps me on the ground, and that is what I enjoy, to work with a small group of young people and to meet young people on these occasions. I will meet 500 or 600 of them next week in Lindau. There are these Lindau meetings of Nobel laureates. Great.
Great. Back to you, Professor Yunus.
So I was not as lucky as you were. I was actually the joke of the world. The process that I just described, the translation of the genetic code, was really very interesting. There was a lot of work done on it. When I stepped in, almost all the details were known, but it was not known how it happens. And for this we needed a structure. And several much more experienced people than me, and much more distinguished, like Nobel laureate people, Wilson, for instance, Watson, for instance, tried and failed. And I came with a little bit different way of looking. And ask Robert, do you remember how they were laughing at me? I was called the dreamer, the fantasioner, the joke, the village fool. And my real enemies called me the liar, because they couldn't repeat or they couldn't do it. So I had to go through it, and also to introduce several advances in the procedure. But we knew that we are progressing slowly and very difficult to explain. So we continued. And I say we because I had two groups of young people. And as Robert, this is what I enjoy until now. So in my case, the beginning was not easy. The beginning mean 16 years. And I think that I was lucky. Maybe you want to hear my luck. I had a bicycle accident and got a brain concussion. And because of it, I couldn't come to an experiment that I planned in Berlin. And I had to go to surgery, actually 2 surgeries. And while I was recovering, I couldn't read science. It was too much for me being so weak. So I read many things, including a report of a delegation that went to the North Pole to see how the bears are manipulating the winter. What is their way of life? What is their metabolism when they, you know that the bears are sleeping during the winter in the North Pole. what we call sleeping. So they wanted to see what happens there, and they found one finding that they thought it was not important or not very important, is that the ribosomes in their cells, inside the cells, on the membranes inside, are organized like these tiles here. And what people couldn't do is to organize ribosomes in crystals. As you heard, crystals are something very, very exciting and very, very ordered. So I thought, if every bear, every winter, every cell has this, it must be a mechanism of nature that people don't know yet. And in my opinion, this was the way to keep the ribosomes from falling apart. I told you earlier they have many, many components. And when ribosomes are being prepared, until today, even while preparing them from the cells, it can be bacterial cells, yeast cells, animal cells, doesn't matter. They fall apart partially. Not all of them function. And I thought, it's not possible that something that is partially fell apart, partially ordered, partially not, it will not crystallize. So first of all, we have to find out to keep them not from falling apart. If bears are having this mechanism because temperature is going to drop, this means pressure. So I thought we can't learn about ribosomes and go to the North Pole and kill bears. This is not the way I wanted to go. But I looked for other pressures. And luckily, I'm living in Israel, and the Dead Sea is in Israel. And although it's called Dead Sea, there are two types of bacteria there. Temperature in the summer can raise 50 to 60 degrees, and the salt is so saturated that it comes out. So I thought this is really pressure. If something lives under these conditions, should be much more robust than normal bacteria. There are only two types of bacteria in all this dead place, and I isolated its ribosomes, and they crystallized. And this is how I started.
So in your own home as well?
Almost in my own home.
So home country.
The lab is a little bit further.
Now I want to open this up to who feels the most strongly about answering this, but do you feel that in 2017 there is equal opportunity for students around the world? Let's say it's Israel, let's say Germany, United States. I mean, I know a lot of people are going and studying, and are they coming back to their country? What do you think about that, Professor Huber?
I perhaps would add to what Ada said. And so we are living in a fantastic time now. So I would even say that none or very few of the methods that we are now using in the life sciences were available when I was a doctoral or a postdoc student. So there was not even protein sequencing was possible at that time. Gene sequencing, we didn't know what genes were at that time, and now it's routine, and thousands and millions of genes are being sequenced, and then the imaging methods. It's fantastic right now what we can do. So your question about the possibilities that students have. Well, they have available these wonderful techniques right now. So well, they have to find out what they really want, what they like. And I would like to encourage them to focus on, to study natural sciences, life sciences in particular. Many of them, unfortunately, for my taste, study law or economics. This is also important, but not what I believe is what mankind is needing now to solve the basic questions, which is food, which is energy, which is health. All of these are fields that require science and research. Mm-hmm. And the fantastic thing with science and research is you never know the outcome. I mean, it's like well, it may be around the corner, or you can have the picture of being in a room and opening a door to a dark room. You don't know what is there. Mm-hmm. Well, this is the feeling that research and scientists say, oh, I have another picture, that of a medieval sailor sailing on the ocean and not knowing where he will land. Mm-hmm. And then finding an island, you see, and entering the island. Nobody has seen that before. So this is the feeling I do have very much in my research.
And obviously this adventure is continuing. Would you say, Professor Serhausen, that after winning the Nobel Peace Prize, what doors then did that open for you? I think our audience would love to know. Well, I mean, you've reached the top, and
it's easier to get access to many things after you win this prize, that's for sure. But let me just add something to what you asked before. Mm-hmm. In my opinion, there are no equal opportunities on the global scale for the young people right now. If you open a position at your place and you see that you get hundreds of applications from India, from Pakistan and some of the other countries, you can easily imagine that there are very different opportunities. I'm traveling a lot and I see a number of laboratories and see in which devastating state, at least in some parts of the world, those places are and how difficult it is for those people to find a suitable position. So in my view, equal opportunities exist in some of the privileged parts of the world, but unfortunately not in many other parts of the world as well. And we have to work on this quite heavily. Now, coming back to the Nobel Prize, I'm frequently being asked, what does one have to do to win the Nobel Prize? I think the most easy answer is, first of all, you have to get old enough and not to die too early. That's one of the major points. But in addition, of course, there are a few other things which I feel are important. I guess it's important to think a little bit, as we say in German, querdenken, which means a little bit thinking transversely to some degree, and being aware a little bit of fields which are not directly related to your own activities. Another point would be, don't trust too much the dogmas which exist in medicine and biology. And sometimes, it's even the worst part, don't trust always your peers. I mean, sometimes your peers give you the worst advice for some reasons, particularly if you start something entirely new. So these are just maybe the last point is you have to have what we call, again in German, a stiff neck, ein steifen Rücken, which means you have to work on a subject for long periods of time and being not so easily distracted by criticism on the right and the left side. That is important, probably because I originate from an area in Germany where people are supposed to be particularly stiff in that respect. That's one of the reasons why we succeeded in the end. But anyway, so these are just a few things which come up. I do not really, I don't think that they relate completely to your question which you ask.
No, it's great. Now, but Professor Primorac, you have brought science intellectuals from 42 countries to this conference, and you are traveling all around the world as well. I mean, what a great opportunity to bring the whole world to one place, to here in Croatia. And I mean, do you feel that you see that equal opportunity through involving so many people? I mean, I think that's great.
Yeah, well, that's exactly what we are trying to do. I think without boundaries, that's our mission. I mean, I spent quite a few years in the States finishing my PhD. Without that opportunity, I wouldn't be able to do things. I have so many students actually sitting here today. I mean, without my education in the States, I wouldn't be able to start with other people from different US agencies. Very important project for Croatia and from the entire world. Identification of the skeletal remains from mass grave. You know, there's so many things that we started because I had opportunities. And only my goal is to all these young fellows and professors, teachers, to help them. And if you ask me about Croatia, during the short period when I was able to change things in Croatia, my priority was to introduce in every single Croatian school 2 foreign languages. So that's what's happening as mandatory. I wanted to make informatization of the all Croatian schools. Then we have something very important that actually I study from my Israeli colleagues. It's actually from the brain drain to the brain gain project. How to attract people to come and to live in Croatia. Indeed, there's a process. Small countries such as Croatia need really help and support from others, but this event particularly is showing you that those things are possible. We are witnessing that today. Indeed, you know, again, I have to tell you, I have to thank dear friends who help us to make this as possible. I mean, as you know, the personalized medicine, it's one field that we are just analyzing, but at the same time, we are touching during this conference other important things. The role of the genetics and forensic science with the goal to make entire society more secure. It doesn't matter if somebody committed crime right now in the States, if we have that person DNA database, we are able to match him tomorrow in Croatia. There's other lectures where we are for the first time discussing out of Africa migration that's happened 120,000 years ago. And for a long time, we believe that Neanderthal men just passed by modern men. Right now, we know that they met. There's about 3 to 5% of the genome of the Neanderthal man today in the Homo sapiens, of anatomically modern man. So there's so many things and opportunities that we are facing today during this conference that make me incredibly proud. Especially, you know, the Dubrovnik, it's a very, very unique place with a lot of culture, tradition. So basically the message that we are sending to the world today from Croatia, yeah, everything is possible if you know in which direction you are going.
Yes, of course.
So I think it would be important to mention here prevention as well. Because in most societies, in Western Europe, in the States and everywhere, there's less than 3% of the expenses for medical research is spent for prevention. And it's always better to prevent a disease than to heal it subsequently. And for those reasons, I think this is really underemphasized completely in our societies. And I'm a very strong advocate for more support for prevention, particularly also in countries like this one here.
Well, I think it's a great time to now get our audience involved. We have some selected individuals who have some questions for our laureates. So please introduce yourself.
My name is Jelena Šarac. I'm a biology postdoc from the Institute for Anthropological Research in Zagreb, Croatia. I have a question for Professor Huber. So, Professor Huber, as a Nobel Prize laureate, you are most definitely one of the leading world scientists. But as we all know, all scientific careers have their ups and downs. So what would you say was your biggest scientific challenge, crisis, or even a failure that you experienced during your career, and how did you deal with it? Thank you.
Well, I mentioned already that research is full of failures, and we have to try to minimize them, but it's probably a little more than 10% of the attempts are successful. There was one thing that Harald mentioned, looking at your peers. I actually had an important happening with one of my peers being able to correct him. The person was a very famous scientist, Adolf Butenandt, the chemistry Nobel laureate. So I worked at that time on a small molecule, an insect hormone called ecdysone. So if you open biology textbooks and you find ecdysone, their structure was unknown. Now this was a time in the early 60s when all of the methods that we now have, NMR, mass spec, were unknown. So the question was already about the molecular weight of it. I was able to crystallize that and in that way determine the molecular weight and found a value that was double of, twice that the Nobel laureate Adolf Butenandt had published.
I don't argue against any type of peers.
So my problem at that time was
peers' advice.
My problem at that time was, should I believe? I mean, it can't be that this famous scientist makes an error and I'm right. And so I repeated the relatively simple experiment over and over again. Suddenly, like a strike from heaven, it was clear to me I was right and this person was wrong. So for me that was really a decisive event. I decided then I could achieve something in science, and I stayed in science and research and in the academia. So that was this an up or a down? I mean, many downs and ups came later than when I had a research group failing to crystallize a material that we wanted to look at. So it's full of ups and downs. You shouldn't be discouraged by the downs. Continue, but there may be a situation then when there's obviously no way to solve the question you posed to yourself. And then you have to stop, which is heartbreaking, you see, because you invested so much work in that, but you have to stop, give up, start something new. That also is important.
And Professor Yunus would also like to respond to that.
Downs, I don't want to talk about only failures. Downs may be even not a failure, just that something doesn't work. It can be heartbreaking. But at the end of the story, most of the downs are opening new ideas, new thinking, new procedures, new way of dealing. So they are, from this point of view, they are bringing ups, or at least advancing. So I can talk about one of our stories when we already had crystals and a little bit of diffraction, not what we expected, maybe 10% of it. We found out that we can collect data only from one exposure, and we needed 360 degrees or part of it if there is symmetry. So less than 1/10,000 of what we needed. And then the crystal disappeared, died, did not diffract anymore. So all crystals do not diffract, but not so fast. This was specific to ribosomes. We repeated 100 times. To get 100 crystals at that time was a work of more than a year for 2 or 3 people. And we repeated it in many synchrotrons in the world, in Europe, in America, in Japan. We got the same thing. This was a minute that we almost stopped. We thought in the beginning that we climbed an Everest, we got crystals that nobody else got, and then we found out that there is an Everest that is higher behind it. So we had to think of what to do, and by chance I met an American professor, actually from Norway originally, Håkon Hope, and he told me that he works on structure of explosives, materials that explode. And he crystallized them, and he got crystals. But when he wanted to bring it to the beam, it exploded. He usually works at cryo temperatures and very, very low temperatures, so there will not be motions within the crystals. So it takes less than tenths of a second, and the crystal exploded, like he had a little bomb. And he gave up this project until he had a student that came and worked earlier in a machine shop, a car repair shop. And he told him that the oil used in gear, the heavy-duty gear oil, is opaque to air. So if he covers his crystal with this oil, it will not see oxygen, so it will not explode. Can you imagine? Looking at my crystals, I thought, using this suggestion, my crystals that grew from the Dead Sea with so much salt and so fragile we couldn't even touch them, to cover them with machine oil, heavy-duty gear oil. This was the second time in my life. Everybody told me it won't work, but it worked. And it worked so wonderfully that afterwards 80,000 new structures were determined under these conditions. With this, we don't use the oil anymore. We found something to replace it, but with this idea. So if I didn't have this problem, 80,000 structures, maybe 75,000 of them wouldn't be made. Yeah. And all of them are, or most of these structures, not by us, by the whole world, are of very high importance, either in new materials or in new medicine important compounds. So I don't want to call it failure, but down, sometimes it's really up.
Yeah. All right, we'll have our next question.
Okay, hello everybody. My name is Mihail Plechko and I'm coming from Zagreb School of Medicine. I'm really honored to be here today and I would like to address my question to Professor Zurhausen. So I actually read about your work on the papilloma virus and you said you had...
Could you speak a little bit up? It's a little bit difficult for me to understand you.
Do you hear me better now?
The bells are doing... I come close to you, then it's easier.
Oh, wait, hold on one second.
Oh, yes, the microphone. All right. I cannot, I'm fixed here.
Yeah. We have the bells are ringing.
Should I come closer?
Yes,
okay.
Perfect.
I'll come closer. So actually, about your work on the human papilloma virus, I actually read you said you had sufficient financial support. But I would like to ask you, I've read that you didn't have appreciation from the scientific society.
Many of the peers.
And can you tell me how you felt then? And additionally, can you tell any advice to young researchers how to deal with situations when they don't get appreciated for their work?
Well, it happened probably to many of us as working in science. I mean, at that stage, I was deeply convinced that we were right in the system. So I went to a meeting in Key Biscayne in Florida and presented there for the first time to a larger audience the idea that papillomavirus must be the cause of cervical cancer. We didn't have the full evidence at that stage, only based on the epidemiological considerations. And indeed, that meeting was devoted to herpes simplex type 2 in cervical cancer, and everyone was sure this is the right agent, and I'm a little bit crazy in thinking of another agent. So that was a devastating experience for me, because everyone really scolded me, and they were ridiculing about this idea that papillomavirus were. It took, even then, it took still 7 or 8 years before we could prove the point. And of course, then the situation changed completely from one day, virtually from one day to the other. We've distributed the probes everywhere in the world, and people confirmed it very quickly, except for 2 groups, which took a few more years before they were able to confirm it. But subsequently, everyone did it. And so it's sometimes a difficult period. When we are working right now on red meat and cancer and milk products and cancer, the situation is a little bit analogous. The only difference is right now that some people start to say, well, at one stage he was right with his crazy ideas. Maybe this time he might be right too. So they're a little bit more cautious in the criticism. It's not that much felt right now as it was in the past. But
now our final question.
Stick to things which you believe they're correct.
So hello everyone, my name is Nika Mikic and I'm a high school student from Zagreb, Croatia. I have a question for you, Professor Yunus. I was just wondering, you stated that your lab was a bit away from your home. I was just wondering if your home was ever your lab, and if so, were there any damages done to your home while experimenting and so on?
When you were a child, you were doing some kind of experiments at your home.
Yeah, we're doing experiments at home.
Experiments at home I did when I was a child.
Did it cause any damages?
One of them
to
caused a fire.
Oh.
When I was 7, but when I was 5, it was much more complicated. I fell off the balcony and broke my head. If you think that this stopped my interest in understanding the world, you're wrong. I continued.
It only fueled it.
I continued.
Okay, great.
I hope the house was okay.
Well, it was a very, very short fire.
Okay, good.
What I wanted to see is how fast water, or to compare between the time that water will go through a piece of wool or kerosene. Kerosene is the compound from which benzene is being made. So it's very heavy, and I wanted to see two things, how fast they go and evaporation. And because we were very, very poor, we lived 3 families in a 4-room apartment, and families have children, and people with just one little kitchen, one little toilet for the whole 3 families. The only place I could do my experiments is at the corner of the balcony. So this is the fire. When I did this experiment, my father came to the balcony because my mother didn't let him smoke inside. She told him smoke only on the balcony. So you can understand, I had kerosene and he came with a cigarette. So there was a little fire. But because it was me and my father and only a little wire, we took care of it quite fast.
So then would you be encouraging others to practice experiments in the home? Or?
I encourage everybody to do what is interesting for him or her, what is exciting, what they want to understand, what they want to investigate, not wait until they have a lab or until they don't have a lab. Do whenever they feel, whatever they can. So you already asked me, maybe I will give you my only advice. I have only one advice, especially to young people that ask how to do science, how to become famous, whatever they ask, I have one answer. Don't ask for advices. Do what you feel, what excites you, what interests you, what you think that you want to understand. If you can do it, that's the only way, in my opinion, to go.
So to
Nothing
pave your
else.
own road.
Yeah, just don't ask for advices. Don't trust advices. You heard from him sometimes advices are not good. So, yes, good. I don't even relate to it. Just do what you feel, what excites you, what encourages you, what enthuses you.
All right. Well, thank you.
You should collect opinions of the elder and then make your choice.
Yeah. So listen to others, and then
trying to be politically correct.
If you have time to gather opinions. Yeah. Sometimes it's burning. No time.
Right.
Especially at a conference like this where there's so many minds in one place. I mean, I think you must get as much information as you can and then take all of that to make your own road. So thank you very much for the questions. And on behalf of Croatian National Television and the International Society for Applied Biological Sciences, we'd like to thank our laureates very much for joining us. Special thanks to Professor Primorac, the Mayo Clinic, American Academy of Forensic Sciences, St. Catherine's Hospital, and our wonderful audience today. So from Dubrovnik, Croatia, thank you very much for watching.
Thank you very much.